Good afternoon. This is the last video for Bio 101. Kind of ends the material for the semester. This video on human genetics is going to be about sex-linked patterns of inheritance and chromosomal mutations, basically, are the two main topics in this video. I did not point it out in video 1, but the illustration on this cover page is on the left is a Y chromosome and on the right is an X chromosome, just giving you an idea for what they look like via an electron transmission microscope. Okay, so another pattern of inheritance, in contrast to autosomal inheritance, is X or sex-linked inheritance. And it can either be via a recessive allele or a dominant allele. This slide talks about X-linked recessive inheritance and gives a couple examples. Hemophilia is one I'll use. A failure of the blood to clot properly. In this case, we use a letter X and a letter Y to represent the sex chromosomes, and then superscripting above the X, we either place a dominant letter or allele or a recessive allele. You will notice that sons are more likely to inherit an X-linked condition at a greater probability than are daughters. And it is often the mother who transmits such disorder or condition to her son. So you notice the two examples are an affected father, and none of their children have the disorder. It says affected father, unaffected mother. And then on the right it has mother as a carrier and father unaffected, and there's a 25% chance for an offspring to be affected, and of the two possibilities for sons, one of the two would contain or have the condition. So it's more likely for a son to inherit an X-linked condition than a daughter. X-linked dominant inheritance involves a dominant allele found on the X chromosome. We got faulty enamel as an example. Again, you might notice that a daughter might have a higher probability of inheriting a disorder if it is a dominant pattern of inheritance. In the example on the left, unaffected father and affected mother, there's a 50% chance of offspring also inheriting this condition, and one of the two is a daughter and one of the two is a son of the two. In the right, it has affected father and unaffected mother, and you notice that because the father is affected, he passes that condition on in the X chromosome to his daughters. So there's 100% chance of a daughter being born with this condition if the father is affected with it. The mother being unaffected, then the son who gets the X chromosome from their mother would have a 0% chance of inheriting this condition if it's a dominant pattern of inheritance. So with sex-linked inheritance, you have an X chromosome and a Y chromosome that you put into the Punnett square, and then you superscript either a dominant or a recessive allele above the X chromosome. You do not do anything with the Y because most of the genes or alleles on the Y chromosome are involved in the development of the male sexual structures. There are a number of disorders on the X chromosome. Some that I had mentioned before include baldness, red-green color blindness, and you might notice testicular feminization, several different types of muscular dystrophy, and Fragile X, some symptoms within the autism spectrum, and so forth. So there's a number of disorders that are found at different positions on the X chromosome. So you notice the numbers. The numbers go as distance or linkage. It's kind of a linkage map in a way, showing you, in this case, how far the particular gene is from the centromere region of that chromosome. So going out 28 units from the centromere and then going up 22 units from the centromere region, if you will. Chromosomal mutations occur either to disrupt the structure of a chromosome, or they can change the actual number of chromosomes. So chromosomal mutations are one type of a mutation. The other type is called a point mutation that we don't typically discuss in this class. But chromosomal mutations are often more severe because they disrupt the structure and or number of chromosomes. So I combined 4 types of chromosomal mutations onto this slide. Letter A is when several genes, the letters A, B, C through H are to represent genes, and a deletion is when one or more genes is removed from a chromosome. A B is when one or more genes is repeated one or maybe several times within a chromosome. So a chromosomal deletion removes a gene or genes. A chromosomal duplication repeats a gene or gene several times. A chromosomal inversion, letter C, is when a portion of a chromosome is detached, it flips and then reattaches to the same chromosome. So the order of the genes is reversed in that portion of the chromosome. And lastly, D, a translocation, is when a portion of one chromosome breaks off and attaches to a non-homologous chromosome, one that is different from itself. So you often see that the non-homologous chromosome will have a portion of it that will detach and will attach to that same chromosome that lost a portion. So it's often a reciprocal, what is called a reciprocal translocation. So these are 4 types of chromosomal mutations that change the structure of a chromosome. the actual number of chromosomes can be changed. And this refers to a fifth type of chromosomal mutation referred to as an aneuploidy, where you either have one extra or one less chromosome than is normal for that organism. The cause of an aneuploidy is called a non-disjunction. It usually occurs in anaphase, and the chromosomes do not separate properly. So one of the gametes acquires maybe an extra chromosome and the other doesn't and acquires one less. So non-disjunction, a failure of chromosomes to separate properly, typically in anaphase of meiosis, tends to result in a chromosomal mutation called an aneuploidy. Aneuploidies within the autosomal chromosomes are called trisomies because those chromosomes will not have two, but they will consist of three. Some of the syndromes are called Down syndrome, Edwards syndrome, and Patau syndrome. Trisomy 21 or Down syndrome is probably the most commonly thought of autosomal aneuploidy. There are aneuploidies that occur in sex chromosomes as well. One can inherit two X chromosomes. That's referred to as Klinefelter syndrome, a sterile male. A male can obtain two Y, be XYY. That's called Jacob syndrome. Usually the male is fertile in this particular aneuploidy. A triple X is a female who has a trisomy with the sex chromosomes, 3 X's. And Turner's syndrome is when the female only inherits one X, and the 0 there means there's not a second sex chromosome that's inherited. So 3 of the sex chromosomal mutations have a diploid number of 47, and XO or Turner's syndrome has a diploid number of 45. As an example of an autosomal mutation or aneuploidy, this usually occurs at a rate of 1 to 7 or 800 births, and some of the features are a flat facial structure, slanted eyes, shortness, heart defects, a shorter lifespan, some level of learning disability, etc. It's typically correlated to the age of the mother, especially if the mother is at or above 40 years of age. But the non-disjunction can also occur within the father, and the 2 chromosomes can be obtained from him, and the 3rd in this trisomy obtained from the mother. Usually the 2 chromosomes that are obtained from the mother, and 1 is obtained from the father for the 3 that are inherited. It is possible to undergo genetic testing for a condition such as Down syndrome. One means is called amniocentesis, where amniotic fluid is withdrawn and the cells are cultured and then karyotyped. Chorionic villus sampling, again, takes in fetal cells, cultures them, and then karyotypes them. The most non-invasive procedure is called non-invasive prenatal testing, where blood is withdrawn and DNA from the placental area is examined and karyotyped. There are 3 different sex chromosome mutations. Turner syndrome is a sterile female. Klinefelter's is a sterile male, and they're illustrated in the figure. Jacob syndrome is a fertile male that has a 2n number of 47.